system. We'll keep that open, start presentation. Reset. Give me a moment here. There we go. Antibodies, that's where we left off. So, we've talked about antibodies frequently, especially when it came to the anti-A, the anti-B, and they bind to the antigens on the cells, on the red blood cells, and can cause agglutination or clumping. Another name for antibody is immunoglobulin. And if you look at this word, immunoglobulin, it's a really descriptive term that describes the function and the structure of an antibody. So I think of globulin, generally speaking, that term refers to proteins. And when we think of an antibody is a protein. It's made up of amino acids. And these are small proteins that have an immune system function, hence the name immunoglobulin. But it's kind of a pain in the butt to say immunoglobulin. So we, physiologists and anatomists, have abbreviated immunoglobulin IG, capital I, lowercase g. There are antibodies. There are different regions to our antibodies. Some regions are constant, some regions are changing. These regions are what allow for our immune system to be so robust, particularly our adaptive immune system. So as we look at an antibody, and let's go with bright green here, we're going to have some polypeptides. We have 1, 2, 3 polypeptide chains. And as we're looking at those 4 polypeptide chains, they're going to be riveted together or anchored together. And when we talk about riveting or anchoring these polypeptides together, we're talking about a disulfide bond. It's a special kind of covalent bond that forms between 2 cysteine amino acids. And we have these disulfide bonds. Let's go with red. We have those disulfide bonds riveting those 4 polypeptides together to give us a quaternary structure for this protein. And as we look at our antibody, some parts of the antibody are the business end. The business end is known as the antigen binding site, which is going to be located at the tips of the Y. I like to think of an antibody as somebody holding their hand up in the air, reaching for something. So you're in the Y shape like an antibody, and you have 4 appendages, just like there's 4 polypeptide chains, and then you grab stuff with your hands and hold them up in the air, just like the antibody will grab stuff with its antigen binding sites. Now, these antigen binding sites are going to change from antibody to antibody. This is where we have most of the variation. As we look at our antibodies, we can pre-program or reprogram our immune cells to produce different kinds of antibodies depending on what has activated the immune response. Recall that an epitope is a different region on an antigen that can trigger an immune response. So we can have different flavors of antigens. Each antigen, each flavor, or each epitope of the antigen is going to have a specific antibody that can bind to it. This is not a genetics class, but it's worth emphasizing just the remarkable amount of variation in our antibodies. We are designed in such a way that when we make our antibodies, we take the genes that are responsible for this variable region, or the active site of the antibody, and it's kind of like rolling the dice, and we see what pops out. We're capable of producing over an order of magnitude more antibodies than we have genes for. This effectively means that even though we aren't pre-programmed for an antibody for a specific antigen, because we randomize things so within this part of our genome, we can randomize response to a new antigen and produce a new antibody that we weren't capable of producing initially. Now, there are 5 classes or categories of antibodies. As we look at these 5 categories of antibodies, immunoglobulin G, IgG, is by far the most common. It's the smallest antibody. It's the most common antibody. It's going to be present in blood plasma, And as we look at this antibody, because it's so small, it can cross the placental barrier. It can go directly from mom's bloodstream to baby's bloodstream. And most of the time, that's really good. Most of the time, a mother is going to produce antibodies that fight off pathogens. And that child will have natural passive immunity. That child will be given antibodies from mom naturally and have a little bit of immunity temporarily. We had talked about hemolytic disease of the newborn. Occasionally, mothers are going to produce antibodies that attack the rhesus antigen on red blood cells. Those antibodies that cross the placental barrier with hemolytic disease of the newborn are a special kind of immunoglobulin G. Now, as we look at our next kind of antibody, immunoglobulin A, IgA. Immunoglobulin A, if we really had to just sum it up in a word, it's the secretion antibody. It's the antibody associated with secretions. When you think of immunoglobulin A, I want you to think of mucus. I want you to think of breast milk. I want you to think saliva and tears. All those things that ooze out of orifices in our bodies are probably going to have some immunoglobulin A associated with them. And when we look at newborns, that passive immunity that's given to newborns from breast milk comes from immunoglobulin A that's present in breast milk. And if you've ever had the chance to hold a newborn baby, they're really kissable. I mean, let's face it, babies are cute. And there's actually some good physiological reasons why a lot of people like to kiss babies. When we look at a newborn child, a lot of times, you kiss it like crazy, he or she like crazy, and the mother's constantly kissing that child and exposing herself to the pathogens that surface of the child's body. And then she's exposed to the child's pathogens, she upregulates her antibody production, and then gives that child antibodies through her breast milk that are on the surface of the child's body. So in many respects, this whole physical touch, this physical bonding that occurs between a mother and an infant, and then breastfeeding that infant is going to help give that infant a lot of natural passive immunity to a pathogen that that child may otherwise have been exposed to. Another antibody class is immunoglobulin E. When we think of immunoglobulin E, I want you to think of an antibody primarily that's going to be associated with allergic responses. So if we're having lots of allergic reactions, we're going to have a lot of immunoglobulin E upregulated into our bloodstream. When immunoglobulin E gets to our basophils and mast cells, we're going to have the release of histamine and other chemicals associated with the allergic response. Another function of immunoglobulin E is that it attracts eosinophils. An eosinophil is one of those leukocytes we talked about that's associated with fighting off multicellular parasitic infections. So in addition to immunoglobulin E being associated with allergic reactions, immunoglobulin E will also be associated with parasitic infections. Think of tapeworms or roundworm, et cetera, et cetera. So those are our antibody types. Let's start talking about the lymphocytes. As we look at our, actually, did I skip a slide? There we go. I was going to say, we didn't get to the big one. The big one, IgM, immunoglobulin M, is a pentamer. And as we look at immunoglobulin M, it has 10 antigen binding sites. Because this antibody is so large and has so many antigen binding sites, this is the antibody that's going to be associated with agglutination. It can take multiple cells and cause them to get stuck together. Now, if these are bacterial cells, awesome. The bacterial cells are stuck together. Those bacterial cells can't function properly. Now, occasionally, immunoglobulin M, IgM, is going to cause our erythrocytes to get stuck together. That's when we have a transfusion reaction and we have agglutination or clumping occurring with our bloodstream. So when we look at immunoglobulin M, I want you to associate this antibody with being very large and taking cells and clumping cells together. And then finally, we have immunoglobulin D. Immunoglobulin D is a smaller antibody that's present on the surface of our B lymphocytes, and it serves to help activate the B lymphocytes. So as we look at our immunoglobulin D, different B lymphocytes will have different variations of immunoglobulin D on their cell membrane, on the surface of their cell. And those different variations of the B lymphocytes with their different antibodies will respond to different antigens. This is one of the reasons why, if we have an antigen-presenting cell, it can activate one B lymphocyte, but other B lymphocytes don't respond to those antigen-presenting cells. All right, now let's talk about lymphocytes. As we look at our lymphocytes, guys, gals, there's some broad categories of lymphocytes. The B lymphocytes, the T lymphocytes. Those are the two broadest categories. But there's a whole bunch of subcategories as well. We'll talk about those subcategories during this presentation today. When we look at our lymphocytes, they are the second most common white blood cell in our body, second only to the neutrophils. And as we look at the T lymphocytes and those B lymphocytes, I want you to think of active immunity. Those are cells of the adaptive or the active immune system, part of our third line of defense. The natural killer cells, which are a variation of a lymphocyte, are present in both the adaptive and the non-specific immune system, or the general and the specific immune system. So as we look at those three categories of lymphocytes, let's talk about the T cells. When we think of T cells, I want you to think of the thymus. Our lymphocytes originate in our red bone marrow. Red bone marrow is present in spongy bone tissue. Spongy bone tissue is overwhelmingly present in flat bones of the body. So if we look at the sternum, if we look at the ilium, Ruby. Yeah, I did, sort of. It says there's three kinds of lymphocytes, just like this slide did. Thank you. So as we look at our T lymphocytes, they come from red marrow that's present in the flat bones of our bodies. Now, all T lymphocytes are going to originate in the red bone marrow. They'll travel through the bloodstream to the thymus. And while they're in the thymus of our bodies, there's some epithelial cells in the thymus that help to maturate the T cells, help to promote the development of these T cells. And while we're developing the T cells, we specifically want antigen receptors put on the surface of those T cells. And there's a special kind of antigen receptor that we want on the surface of the T cell. We want this antigen receptor to not respond to a self-antigen. If it responds to something that belongs in our body, it means that T cells are to attack our own body cells or our own tissues. That's not a good thing. The other thing that we look for is that we want those T cells to respond to foreign antigens. So they don't respond to self-antigens, but do respond to foreign antigens. And if those two criteria are met during the maturation of the T cell, that T cell is declared immune-competent, capable of recognizing what does and does not belong into our bodies. And we have positive selection throughout this process. We preferentially will select T lymphocytes that meet these two criteria. And if the T lymphocytes don't meet both criteria, if we don't get a yes and a yes, we kill them off. Hence the name positive selection. Now, within our thymus, occasionally our T lymphocytes don't pass the test. Occasionally, they respond to self-antigens. And as they're being tested, this testing process is going to involve exposing the T cells to the epithelial cells in the middle, or the medulla of our thymus. And if those T cells respond to those epithelial cells in the middle, or medulla of the thymus, we take them out. This process here is referred to as negative selection, in that if we get a no, we kill them off. It's worth mentioning, though, occasionally this process doesn't work. Every once in a while, we pump out T lymphocytes that don't respond to foreign antigens. They're effectively neutered T lymphocytes. They aren't capable of mounting an immune response to something that does not belong in our bodies. And occasionally, we will pump out T lymphocytes that respond to self-antigens. Unfortunately, we didn't have enough time to really dive into immune system-related diseases, but most autoimmune diseases are going to be associated with our T lymphocytes or our B lymphocytes failing this test and not being killed off when they're supposed to be killed off. And then they enter the bloodstream and they start attacking our own tissues. Thankfully, that's not very common. In our bodies, we also have a lot of B lymphocytes. When we think of B lymphocytes, I want you to think of the bone marrow, particularly the red bone marrow. The namesake of our B lymphocytes was the bursa of a chicken that they were initially isolated from, and it just works out really nicely for those of you that like illustration. Red bone marrow, or bone marrow, more generally speaking. So as we get our B lymphocytes, they start in the red bone marrow, and then they're going to mature within that bone marrow, and occasionally they'll be sent out into the body to finish the maturation process in the body. But primarily, think of maturation in the red bone marrow for our B lymphocytes. After the B lymphocytes have maturated and they're immunocompetent, they don't respond to self-antigens, they do respond to foreign antigens, they're going to exit the red bone marrow, go through the bloodstream, and travel to the lymph nodes. Preferentially, these B lymphocytes hang out in the lymph nodes. The reason they hang out in the lymph nodes is because this is where pathogens tend to be concentrated in our bodies. At the beginning of this presentation, we talked about the lymphatic system, how we have extracellular fluid constantly building up in our bodies, and we take the extracellular fluid and suck it up into our lymphatic capillaries and then send it through the lymph nodes to the lymphatic ducts and then eventually drain it back into the venous return, the two subclavian veins that head back to the heart. And as that lymphatic fluid is being sent back to the heart, it'll stop at the lymph nodes and be filtered. While those lymph nodes are filtering the lymph, the pathogens get stuck in the reticular tissues and end up having them slow down for the B lymphocytes to be exposed to them. We also find that our B lymphocytes can patrol other tissues in our bodies, but overwhelmingly in the lymph nodes. Now, within our lymph nodes, let's say we have a B lymphocyte that's going to be activated. And that B lymphocyte can be activated 3 different ways. The least common way, or the hardest way to activate B lymphocyte, is for it to bump into the pathogen directly. B lymphocytes, they don't recognize a good thing when it hits them. I mean, they can't really recognize and respond to the pathogen directly most of the time. It's difficult for them to do that. However, if an antigen-presenting cell, so either the dendritic cell, the liver macrophages, the alveolar macrophages, or maybe the macrophage in general, swallows and phagocytizes the pathogen, decorate the cell membrane with antigens from the pathogen, that antigen-presenting cell can say, hey, pay attention. This is important. Don't ignore me. And will make that B lymphocyte be much more likely to respond to the presence of the pathogen. There's another way that our B lymphocytes can be activated, and that's in response to a T helper cell, a special kind of T lymphocyte that's kind of the coordinator of our adaptive immune system. Once our B lymphocytes are activated, they divide very, very rapidly, and they form 2 kinds of cells that we'll talk about in subsequent slides in more detail. Those 2 cell types they form are plasma cells and B memory cells. An activated B lymphocyte will turn into plasma cells and B memory cells. Now, these antigen-presenting cells are pretty important. They help our B lymphocytes and also our T lymphocytes respond to the presence of an antigen. You can kind of think of the antigen-presenting cell as kind of like the tattletale you knew in elementary school. That person who was always going, teacher, 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 so-and-so had an extra milk carton during snack time. A lot of times the teachers ignored the tattletale, but occasionally the tattletale elicits a very strong response out of the teacher. And the antigen-presenting cells are the same way. They'll bounce around, showing multiple lymphocytes this antigen and say, hey, hey, did you know about this? This is important. I think this is a big deal. Most of the lymphocytes don't have the ability to respond to the antigen being presented, so most of the lymphocytes ignore the antigen-presenting cell, or a specific antigen-presenting cell. Occasionally, though, we'll have the right antigen-presenting cell with the right antigen present that antigen to a lymphocyte that can respond to it, the right kind of a lymphocyte. And that's when we initiate the immune response. We have a lot of different kinds of antigen-presenting cells. All of these antigen-presenting cells depend on a category of proteins called MHC, major histocompatibility complexes. These MHCs are the driver's license, your passport, your visa. They are the ID tag of our body. They are what let us know not only between a liver cell and a stomach cell and a red blood cell, they also let us know the difference between cells that belong in me and cells that came from you. Just because we're humans and have very similar genomes doesn't mean we have the same identification tags on the surface of our cells. When we look at these MHCs, practically speaking, these are very important for donations. When we do tissue donations from a donor to a recipient, we need to match up as many MHCs as possible. And this is a problem because there's so many different kinds of MHCs on the surface of our cells that it's very rare that we get a perfect match for a tissue donation unless we're doing a donation from an identical twin to the other identical twin. As we look at these MHCs, they can be thought of as proteins on the surface of a cell. And these MHCs on the surface of the cell can do multiple things. For instance, these MHCs can hold a little piece of a protein or a little piece of a complex carbohydrate. They can hold an epitope of an antigen on the surface of the cell membrane. This is how an antigen-presenting cell will ultimately display the little bits and pieces of the bacterium that it's phagocytized on the surface of its cell membrane. Now, as we look at this process, this multi-step process involves initially taking our bacterium, phagocytizing it. It's wrapped up in a vesicle. That vesicle fuses with a lysosome and is renamed a phagolysosome. It's a phagolysosome is, practically speaking, a bacteria that's in the process of being digested by the enzymes within the lysosome. As those highly oxidative enzymes rip apart the bacterium is chopped up into little pieces. Most of the pieces are ejected through exocytosis, but some of those pieces are used as decoration or put into an MHC protein and then presented to the T and B lymphocytes. So in our bodies, we'll have T lymphocytes wandering around, constantly inspecting antigen-presenting cells. And if it finds an antigen-presenting cell that only has a self-antigen, that T lymphocyte is going to ignore it. The T lymphocyte will look at that cell and say, oh, you belong here, and then move on and focus on the next cell. Occasionally, though, those cells are going to display a non-self antigen. Now, if we initiate a T cell response, T cells, which are associated with cellular immunity, will start to destroy the cell displaying the non-self antigen. So as we look at this process, let's think about what could cause a cell to be destroyed by a T cell. If a cell is infected with an intracellular pathogen, a virus, intracellular bacterium, that infected cell has the ability to take little bits and pieces of the virus or little bits and pieces of the intracellular bacterium and display them on its surface. And when a cell displays part of an intracellular pathogen on its surface, it's a way of advertising to a T lymphocyte to say, hey, I'm infected, I'm pumping out more pathogens, and you really need to kill me off for the good of the organism as a whole. And that's what will happen. When the T lymphocytes are exposed to the non-self antigen, they initiate that immune response and trigger autolysis within our bodies and kill off those cells. There's also going to be a coordinated immune response. We're also going to start to produce antibodies, potentially if it's a bacterial pathogen. We also can use antibodies to bind to extracellular viruses when those viruses are extracellular. And we're going to also start the differentiation and proliferation of the T cell. Once activated, that generic T lymphocyte will reproduce and divide and start to produce specialized versions of the T lymphocyte. These T lymphocytes, as I mentioned a couple times already, are focused on cell-mediated immunity. In other words, T lymphocytes are really good at killing our own cells. That's what they specialize in. A T lymphocyte specializes in killing off human cells. In particular, we want the T lymphocyte to kill off human cells with that intracellular pathogen, the virus or the intracellular bacterium. As we look at this process, our T lymphocytes, once activated, will differentiate into 4 subcategories: the cytotoxic T, the helper T, the regulatory T, and the T memory cell. So let's talk about those 4 varieties of T lymphocytes. And I feel like this slide, I'm just flying through material. Most of this stuff, when I took immunology, we had a unit dedicated to each of these cell types in our class. So this is just scratching the surface right now. When we think of a cytotoxic T cell, the old school name was the killer T cell. And it's probably, I'll admit, my favorite name, even though cytotoxic is technically a better name because it's toxic to other cells. Cyto meaning cell, toxic meaning deadly or destructive. Killer T cells are going to be the hitmen of our immune system. They're the ones that are going to actively kill off the infected human cells in our bodies. When we look at this verbiage over here, the T8, CD8, or CD8 positive, this is going to refer to the specific kinds of MHCs that are present on the surface of these different kinds of T lymphocytes. This is mostly an FYI. I'm not going to ask you any questions on your lecture exam about the specific MHC molecules on specific kinds of T lymphocytes. But I have to include it just because it's really important. And if you take an immunology course, you'll learn why it's really, really important that different lymphocytes have different kinds of MHCs on their surface. As we look at our helper T cells, or T sub H cells, these are going to be the coordinator, or the CEO, or the planner of our immune system. They are going to run around and activate a bunch of other cell types. Helper T cells directly do not kill off any intracellular antigens. But a helper T cell does upregulate the activity of other T cells, and it will also upregulate the activity of other macrophages and upregulates the activity of other B lymphocytes as well. You can think of the helper T cell as that cell type that causes you to get the maximum possible immune response. We can still mount an immune response without helper T cells, but we get kind of weak, lame, disorganized responses with our immune system. But with a helper T cell population present, we get a much more robust immune response present. We also have regulatory T cells. Regulatory T cells are important because they help to down-regulate the response of our T lymphocytes. If we have too many cytotoxic T cells floating around in our bodies, those cytotoxic T cells eventually will start looking for something to kill and will eventually start destroying off our own human cells. It's kind of like teenagers with too much free time. If there's too much free time for a teenager, eventually they'll find something inappropriate to do. And the same can be said of our cytotoxic T cells. If we have too many cytotoxic T cells in our body, eventually they'll start attacking our own human cells. So we need some way to down-regulate the activity of our cytotoxic T cells. And that's going to be the T regulatory cells. The helper T cell will upregulate cytotoxic T cells, and then we fight off the infection. But when we're done destroying the viral infection, we need to get rid of the cytotoxic T cells. We need to disband the military, so to speak. And that's where the regulatory T cells come into play. A lot of research right now has found that a malfunction of regulatory T cells is also going to be linked to immune diseases or some kind of an immune response where our body starts to attack ourselves. And then finally, we have memory T cells. Memory T cells give us long-term immune memory. Those memory T cells function the same way that memory B cells function, in that they remember that they've been activated. And the memory T cell is kind of like that person who bears a grudge. And once you know somebody with a chip on their shoulder, and they're always looking around, and they're looking for a reason to get angry. You know, you've met somebody like that. Our memory cells are kind of the same way. They're wandering around our body saying, where's that antigen? Where's that pathogen? I know it's somewhere around here. And it's looking for a reason to upregulate the immune response. When it's exposed to that antigen, it will immediately and very dramatically upregulate our immune response so quickly that the pathogen can't make us sick. And this is a primary reason why we very rarely get the exact same illness twice, because we produce the memory B and the memory T cells, and can respond and react so quickly that the pathogen doesn't have time to reproduce and induce a disease state. So, regardless if we're looking at humoral immunity with our B lymphocytes or cellular immunity with our T lymphocytes, there's a generic three-step plan. First, you need to recognize the threat. Then you need to attack or react to the threat. And then afterwards, you need to learn your lesson. You need to remember what that pathogen was so you can respond appropriately the next time. So let's look at the 3 R's of immunity, or the 3 steps of our immune response. Step number 1. We need to have an antigen-presenting cell activate the lymphocyte. That antigen-presenting cell is going to carry on an MHC a little piece of the antigen that it consumed. And that antigen-presenting cell will then bounce into a cytotoxic T cell or a T helper cell and help to upregulate the immune response, help to activate that lymphocyte. Antigen-presenting cells are pre-programmed to head to the nearest lymph node once they have an antigen on their surface. And it's a good thing that they do that, because we have a lot of T and B lymphocytes hanging out in the lymph nodes waiting for an antigen-presenting cell to activate them. And this is kind of an awkwardly placed review question. I'll cover that. But the kind of immunoglobulin with best description associated with its function. So we have those immunoglobulins, those different kinds of antibodies that are being produced. Which is which? Make sure you spit your answer down to 5 seconds. And time is up. So, guys, gals, as we look at these antibodies, IgG, immunoglobulin G, is going to be the smallest and most common antibody in our bodies. IgA is the one that I want you to associate with secretion, so associate it with mucus, breast milk, saliva, tears. When we look at immunoglobulin M, that's the pentamer. It's made of 5 subunits. It's the biggest, largest antibody within our bodies. And then finally, we have immunoglobulin E. That's going to be associated with allergic responses and multicellular parasitic infections. See, when we respond. Now we're starting the react phase. And during this react phase, we start the differentiation of our T lymphocytes, or our lymphocytes. And this is going to occur through co-stimulation. First, that antigen-presenting cell exposes the lymphocyte to this antigen. And then the antigen-presenting cell needs to give it a elbow on the side. This extra nudge to say, no, really, you should respond to this antigen is called a co-stimulatory protein. And when we see this process, it's represented by antigen as the little yellow dot. There, the little yellow one is the antigen. And then the red line represents the co-stimulatory protein. To activate our T lymphocytes, we first need to expose the T lymphocyte to the antigen. And then after exposing to the antigen, it's exposed to the co-stimulatory protein on the antigen-presenting cell. Another way you can think of this co-stimulatory protein is it's a way for the T lymphocyte to double check. Okay, is this antigen I'm binding to on an actual antigen-presenting cell that belongs in this body, or is this some other random cell or pathogen that I'm binding to, and it's not being presented to me by an antigen-presenting cell? Or you could think of this antigen-presenting cell as, or the co-stimulatory protein, as a way to verify that, yes, an antigen-presenting cell is telling me to respond. After we have presentation and co-stimulation, we have reproduction. This is known as clonal selection, this process of the lymphocyte reproducing many, many times over. We go through mitosis and cytokinesis and duplicate the nucleus and the cytoplasm many, many, many times over. And you probably all heard that old story about how somebody asked to get paid a penny a day and have their pay doubled every single day. And by the end of the month, they're getting paid millions of dollars a day, or just a ridiculous number of dollars a day. Because if you double and then double and then double, you exponentially increase the number you're working with. And that's what happens during clonal selection. We double the number of T lymphocytes again and again and again and again after we've activated them. And we end up with millions upon millions of these lymphocytes that are going to respond to that very specific pathogen patrolling our body. Now, as we are reproducing our T lymphocytes, we start to differentiate them. We start to delegate different purposes to different T lymphocytes. Some of those T lymphocytes are going to be cytotoxic T lymphocytes, or killer T cells. They're going to be the frontline soldiers going around trying to kill off the cells that are infected with the virus or the intracellular bacterium. Sometimes they'll differentiate into the helper T cells, which recruit other cells of the immune system. Both the cytotoxic T and the helper T cells are referred to as the effector cells because they cause the immediate effect of removing the pathogen from our bodies. And then we have the T lymphocytes that are the long-term members. Those are the T memory cells that are being produced. And those cell types function primarily to help us respond quickly the next time we're exposed to that pathogen. And then we have the respond stage. And we're still in that response stage. We need to attack. And part of this attack process is going to involve killing off cells with intracellular pathogens. And in this process, the T lymphocytes need to recognize the antigen MHC complex. In other words, these T lymphocytes need to find cells with an intracellular infection. And when these T lymphocytes find a cell that are presenting an antigen on their surface, a non-self antigen on their surface, they respond by secreting a kind of chemical known as interleukins. These interleukins are going to have three effects. The three effects include recruiting neutrophils and macrophages to phagocytize and destroy any bacterial cells or infected human cells. We're also going to stimulate the attraction of natural killer cells to both attack bacterial cells directly and attack human cells that are infected. And we're also going to stimulate the production of more T and B cells. So when we look at these interleukins, those interleukins are a very powerful category of chemicals that upregulates our immune system response. There are some avenues of research right now that focus on giving patients artificially synthesized interleukins to upregulate their immune system when they have a lack of T or B lymphocytes. Another aspect of this is that the cytotoxic T cell needs to bind directly to the surface of the infected cell. And when that cytotoxic T cell binds to the surface of an infected cell, it will recognize non-self antigen being displayed in an MHC molecule. And when it recognizes something that does not belong, the cytotoxic T cell kills off the infected human cell. And it will kill it off a couple different ways. First, it can release perforins, or perforins, depending on potato, or it can release the granzymes. These are the same kinds of chemicals that are released by the natural killer cells, and will kill it off in the same way. So the perforin is going to perforate the surface of the human cell. The granzymes are going to be proteases that digest the proteins inside of the human cell, making those enzymes we need to maintain our cell's metabolic activity non-functional enzymes. Our cytotoxic T cells are going to release more interferons. Those interferons are going to reduce the ability of viruses to attack neighboring cell types. The infected cell itself also will release the interferons, but the cytotoxic T cell, in addition to the affected cell, will be able to release those interferons. And recall, interferons don't help the victim, don't help the cell that's already infected, but interferons warn neighboring cells, the cells next door, and will say, hey, whoa, there's a virus here. You need to look out for this specific virus. And it will confer temporary immunity to the virus for those next door neighbor cells. And then, as we look at the cytotoxic T cells, they also can release something known as tumor necrosis factor. Tumor necrosis factor is a specific kind of chemical that destroys cancerous cells in our bodies. So when we look at a cancerous cell, most cancerous cells in our bodies don't have non-self antigens being displayed on their surface. However, they're still malfunctioning. The MHCs on the surface of a cancerous cell don't look quite right. It's kind of like a false ID. And this false ID, or fake driver's license, it can fool some of the lymphocytes, but it doesn't fool all of the lymphocytes. And when a lymphocyte recognizes a fake ID or a cell that is cancerous, it kills it off right away or responds very aggressively. An active area of anti-cancer research right now is looking into synthesizing variations of tumor necrosis factor so that our T lymphocytes and our macrophages can better respond to those carcinogenic infections or the cancer cells. After our T lymphocyte has destroyed that one particular human cell, it then looks for the next cell, the next infected cell in our bodies. And it'll keep going from infected cell to infected cell and look for a human cell to kill off. These T lymphocytes are pre-programmed to try to kill off human cells. Now, after we've recovered from the infection, we need to have some kind of immune memory. And this comes from our memory cells. We have B memory cells that give us humoral immune memory, or help us to have immune memory to bacterial infections. We also have T memory cells that give us immune memory primarily to viral infections. And as we look at these memory cells, they are very long-lived. They can live for months, years, or decades, depending on the kind of infection that we're looking at. And because these memory cells are so long-lived, and in particular, so numerous, the next time we're exposed to that pathogen, we can activate our immune response much quicker because we have more cells in our body actively looking for that particular pathogen so they can respond aggressively. Once our memory cells are activated or re-exposed to the pathogen sometime later in life, we have such a quick response that most of the times we don't even notice that we had that pathogen. We don't even notice the symptoms of the disease. So that was cell-mediated immunity. We also are going to have humoral immunity. And when we look at humoral immunity, this is going to be associated with extracellular pathogens. Or another way to think of this is when we think of humoral immunity, I want you to think of bacteria or bacterial infections. Humoral immunity is going to be localized or primarily mediated by B lymphocytes, and will focus on attacking the bacterial cell directly. And just like with cellular immunity with the T lymphocytes, when we look at humoral immunity, it's going to have the recognize, the respond, and the remember, or those three stages of response. So let's look at how humoral immunity differs from cellular immunity. How do we handle bacteria versus viruses? As we're looking at the recognition phase, it's going to be different from the recognition phase of our T lymphocytes. We're going to have, on the surface of our B lymphocytes, lots of antibodies. And those antibodies, or antigen receptors, on the surface of the B lymphocytes allow for those B lymphocytes to respond or bind to antigens directly. A lot of those antigens will be associated with a specific pathogen, and when those antigens bind to the receptors or the antibodies on our B lymphocyte, they're going to be phagocytized or brought into the cell through receptor-mediated endocytosis. That's something that we had like maybe 1 or 2 PowerPoint slides on last semester. So let's review and refresh a little bit. Receptor-mediated endocytosis is a way to bring large molecules into the cell that normally won't get in otherwise, and it concentrates and purifies those molecules before bringing them into the cell. So if we have only one antigen bind to the surface of a B lymphocyte, generally speaking, it's not going to be endocytized or brought into the cell. But if we have a lot of antigens bind to the surface of the B lymphocyte, they'll be concentrated together on the surface and then endocytized or brought into the cell. After it's brought into the cell, it's digested. Is that in the hallway? Okay. Wow. Whoa. It's like sliding the models across the countertop in lab. Oh. I will never get used to that noise. So where was I? Irritating noises. B lymphocytes. Okay. So as we look at our B lymphocytes, they phagocytize the antigen, they digest it, and then they take little bits and pieces of the antigen and display it on the surface. They act as an antigen-presenting cell. And as that B lymphocyte that's digested the antigen presents it on the surface, it's going to take that antigen that's digested and show it to a T helper, or T sub H lymphocyte. And it's going to say, hey, look what I found. I think you're interested in this. And the helper T cell will generally go, whoa, this is a big deal, and then run around and activate other cells in our immune system to coordinate the immune response. So after the B lymphocyte is acting as an antigen-presenting cell, is that a hand up in the back? Okay, just stretching, all right. After that B lymphocyte that's acting as an antigen-presenting cell will activate the helper T cell. That helper T cell releases a bunch of interleukins. Those interleukins serve to activate both the B lymphocyte and upregulate T lymphocyte production as well. Once the B lymphocyte is activated it goes through clonal selection just like the T lymphocyte does. In other words, it makes genetically identical copies to itself. It doubles, and then it doubles, and then it doubles, and we have that positive exponential increase of the population of B lymphocytes. Most B lymphocytes are going to turn into plasma cells. And when we talk about plasma cells, they are the cell type that make antibodies. Plasma cells make antibodies. I'll say this a third time. Plasma cells make antibodies. So when we think of those 5 categories of antibodies that we talked about, they're all made by plasma cells. And it's not just that plasma cells make antibodies. Plasma cells make a ginormous amount of antibodies. Once they're up and running, once the production lines have been built, we're talking about 2,000 antibodies per second per plasma cell being pumped out into our bloodstream. It's a ridiculously, mind-bogglingly large number of antibodies. Now, as we look at these B lymphocytes, they release the antibodies. Antibodies have their active sites that will bind to antigens. If it's IgM, or immunoglobulin M, that antibody will cause multiple bacterial cells to get stuck together so they can't effectively move around and infect other cells or tissues in our bodies. If we're looking at immunoglobulin G helps to opsonize or make a cell be ready to be phagocytized. I want to say it was Tuesday of last week. We talked about this process about how if a bacterial cell is covered with antibodies, that bacterial cell is preferentially going to be phagocytized by the macrophages and dendritic cells, other antigen-presenting cells of our bodies. So some of the antibodies disable the bacterial cells. Some antibodies make those bacterial cells be primed for phagocytosis. Other antibodies, when they bind to those bacterial cells, are going to trigger the complement cascade so that those membrane attack complexes can start to form in the cell membrane of those bacterial cells. So these antibodies are going to do many different things. They're wonderful for us. It's worth mentioning, though, antibodies do not kill bacterial cells directly. Antibodies help other aspects of our immune system kill the bacterial cells more effectively, but they don't do the killing directly. So we have the B lymphocyte. An activated B lymphocyte turns into a plasma cell that makes the antibodies. And then some of those activated B lymphocytes that are produced from clonal selection are going to turn into memory B cells. They're very similar to memory T cells in that they are going to allow for us to have an active immune response. So just to reiterate, the antibodies don't destroy the cell. The bacterial cell antibodies preferentially highlight a cell to be destroyed or tag it for destruction. B memory cells are going to be the long-lived cells of our immune response associated with humoral immunity, or immunity for extracellular infections. Let's talk in more detail about how antibodies can kill, or not kill off directly, but coordinate the destruction of our extracellular pathogens. We can have agglutination. We've talked about this one already. Agglutination is when antibodies bind to the A antigen, the B antigen, or the rhesus antigen on the surface of a red blood cell. There are some other antigen types, like the Bombay antigen, but those are very rare worldwide, represent a fraction of a percent of the human population. We'll focus on the biggies. So as we look at those immunoglobulin M's, those really large antibodies, they can cause multiple red blood cells to get stuck together or clump together. And this big chunk of cells represents a very enticing meal for a macrophage or some other phagocytotically active cell. It's kind of like the difference between a whole and a big, or like, I know it was called the yolk. It's basically a giant circle about the size of a pizza. It's very attractive looking. If you ever go to Ripon, get one of those things with a friend. Don't eat it by yourself. And the same can be thought of as those immunoglobulin M's. They take those bacterial cells and put them together to make a larger, more appealing target to be phagocytized compared to phagocytizing one individual measly tiny bacterium at a time. Occasionally, when we have a transfusion reaction, we have clumping of our red blood cells. That's a bad thing. Thankfully, that's the exception. And during this process, we're going to tag the cells for those protein cascades that we talked about earlier in this presentation to help activate cytolysis through membrane attack complexes, or tagging the cells to be more likely to be phagocytized. One of the key differences between the agglutination and the complement fixation, even if a cell is covered in antibodies, it's still more likely to be phagocytized than a cell that's not covered in antibodies. Let's back to the analogy. We can have a regular donut hole, or we can have a really big donut. And most of us like big donuts. But if we had only individual from, I don't know about you, but I'm going to choose the donut hole covered in sprinkles because it's more delicious tasting. In the same way, an individual bacterial cell covered in antibodies is kind of like that sprinkled covered donut hole. It's more attractive than the plain one with only powdered sugar on its surface, and is more likely to be phagocytized. We also have precipitation. Precipitation is different, distinctly different from agglutination, because agglutination causes cells to stick together. I'm going to emphasize this. Agglutination causes cells to stick together. Precipitation causes individual molecules to stick together. Practically speaking, this precipitation is what happens when we are given antivenom. Let's say you have a snake bite. There's some molecule in the snake that's toxic to the body. We're given antivenom. The antivenom contains antibodies that bind to that particular toxic molecule and cause that toxic molecule to precipitate our food as opposed to attacking the cells of our bodies. And then finally, there's neutralization. A bacterial cell that's covered in antibodies has a very difficult time functioning. It can't do the things it normally wants to do. It's kind of like handcuffing it or tying its hands behind its back, so to speak, in that it can no longer get into the tissues that it normally would get into because it's being choked and clogged and bogged down by all of these antibodies binding to the antigens on its surface. Let's look at our immune response. The first time you get sick is always the worst time. Most of us remember that first time we were exposed to the mononucleosis virus. I know there's a couple students here that talked about how they have mono right now. I remember when I was 24, that's the first time I got mono. It hit me like a ton of bricks, and for about 2 weeks, I couldn't do anything. Because my primary immune response, my immune system had never been exposed to that before, and consequently, when I was trying to respond to it, it took a while. My immune system had to get its act together. And while my immune system was going, hey, I think I should do something about this. Are you sure I should do something? Yeah, we should do something about this. Well, for lack of a better analogy, my immune cells were trying to figure out what to do. That pathogen was rapidly reproducing in my body and reproduced to the point that I had the disease state set in. Eventually, though, our immune system figures out what it needs to do and will respond to that pathogen. And eventually, we'll have the multiplication of the B cells. The immune competent B cells are activated and turn into plasma cells and B memory cells. And the plasma cells and B memory cells are going to pump out 2 key kinds of antibodies in high levels: immunoglobulin G and immunoglobulin M. When we think of immunoglobulin M, that's the antibody that functions for agglutination, that's going to take those bacterial cells and cause them to get stuck together. And when we think of immunoglobulin G, that's the antibody that helps with opsonization, to make those individual bacterial cells more likely to be phagocytized. So first, we'll have a peak in immunoglobulin M, and we'll start to bind to and neutralize individual bacterium and cause them to be clumped together. And then we have a peak in immunoglobulin G to opsonize the individual leftover bacterium. And let's look at the timeline. This takes a couple days to have our initial response. So we have initial exposure or infection. And we're looking at about 4 to 5 days, depending on how robust your immune system is, before you make any antibodies, before you do anything about this infection. But you get sick, you suffer through that first infection, and now you're going to have some B lymphocytes left over, some memory B cells left over. And those memory B cells are wandering around our body, just itching to get in a fight with that pathogen, that exact same pathogen again. And the next time they're exposed, we call that the secondary response. Before we talk about the text, I want to focus on the graph. Instead of days, 4 to 5 days for our response, we're looking at 4 to 5 hours for our secondary response to be activated. In other words, those antibodies are going to be pumped out nearly instantaneously, biologically speaking, in response to re-exposure. And instead of having just a little bit of immunoglobulin M and a little bit of immunoglobulin A, we have incredibly high concentrations of immunoglobulin G being pumped out nearly instantaneous, in less than a quarter of a day, after we have re-exposure. This is a little bit of an older song. It was a popular song when I was in high school. I think it was called Harder, Better, Faster, Stronger. And it really epitomizes the secondary response. You hit the bacteria harder, better, faster, with a stronger response. Now, this response is so rapid that the bacteria don't have time to reproduce, don't have time to make us sick, and you don't even notice that you were infected. You are infected without a disease state setting in, which is pretty cool. That's how most of us like to be infected. You know, you get the infection, but nothing bad happens. Now, it's worth emphasizing that these memory cells last a while, but they don't last forever. Some pathogens are easier to recognize than other pathogens. A notoriously difficult pathogen to recognize is the tuberculosis pathogen. It's a bacterium that's covered in a waxy capsule, and that waxy capsule on the surface of the TB bacterium makes it very difficult for us to recognize and respond to that bacterium. Other bacterium that are truly gram-positive or truly gram-negative, they always are going to have the same antigens on their surface, and we get a better, more robust response to those bacterium from our immune memory. Another thing to think about is where the bacterium is going to be located in our body. These antibodies are for extracellular pathogens. If that bacterium is hiding inside of a cell in our bodies, it can't be touched by the antibodies. And it can be free to reproduce inside of our own human cells as long as it's not as long as it's not lysed and having bits and pieces of it presented on the surface to trigger a response from the natural killer cells or the cytotoxic T cells, which specialize in killing intracellular pathogens. And that was our crash course on the immune system. So we took a huge topic and squeezed it into one chapter of our class. Just to reiterate the broad points of our immune system, we have 3 branches of our immune system. Our first defense is nonspecific and outside of our bodies. think of the skin and our mucous membranes. A key component of our skin is going to be the acid mantle of our skin that makes our skin slightly acidic and highly saline. We also have the stratified squamous epithelium of our skin that's keratinized that makes it very difficult for bacterium to get into our bodies. Inside of our bodies, in our respiratory and digestive tracts and reproductive tracts or genitourinary tracts, we have a lot of mucous membranes that will help to prevent bacterial and pathogen access. If something gets past the first line of defense, the extracellular nonspecific defense, there's our second line of defense. That second line of defense is going to be inside of our bodies and nonspecific. That's going to be things like the macrophages, phagocytizing anything that doesn't belong, the neutrophils, indiscriminately destroying bacteria. We're going to have the fever response. We can have the opsonization and complement cascades, all being aspects of the second line of defense inside of our bodies that are nonspecific. Linking the second line of defense to the third line of defense are going to be those macrophages and other antigen-presenting cells. They phagocytize something generic. They're like, oh, this is something that doesn't belong in our bodies. Let's chew it up, swallow it, and then show bits and pieces of what was left over. And then we'll show it to other cells of the immune system. Those cells of the immune system are immunocompetent T cells and B cells. And when those cells are activated, that triggers the third line of defense, the adaptive or specific immune system. And then we'll have T cells reproduce and specialize in killing intracellular pathogens, or we'll have B cells reproduce and specialize in killing extracellular pathogens like bacteria. So that's all I got for you today. We have a little bit of time left, but I don't want to jump into talking about the respiratory system. We'll just call it early today.